Alright, today we're going to talk about cell controls and on cell division, how we control the cycle of the cell. Okay. So, cell regulation is very carefully controlled. Not all of your human cells divide at the same rate. Muscle and nerve cells, once they develop, they don't really divide at all. Your skin, your digestive tract, your bone marrow, though, divides really rapidly every few hours throughout your entire life. So the body has to keep these under control, because if it loses control of the cell cycle, then it creates problems such as cancer. Okay. So cells will usually stop growing until they contact each other, or keep growing until they contact each other. Okay. It's called contact inhibition. If an opening appears, like I cut my skin, the cells are going to continue to grow, divide, and replace themselves until that skin is covered. And we call this contact inhibition. Once it gets covered, once all of these cells are touching, they stop growing. By the way, here the cells are, this is one cell. You can kind of see where the cytoskeleton is. This is a nucleus. So as these cells, here's a cell, here's a cell. As these cells build and touch each other, they stop reproducing, they stop going through mitosis. Until if I were to cut a swath through here, and they're no longer touching, they'll start dividing again, start making new cells until they fill that gap. Okay. Cyclins are the proteins that control the cell cycle. This is one thing that's very nicely named. Hey, if you take the cytoplasm out of a cell in mitosis and put it into a normal cell, it makes that normal cell divide. It's kind of cool. It tells us there's those proteins in the cell that control the cell cycle, and they're specifically in the cytoplasm. There's both internal and external controls, and we'll talk about each of those. Okay, so some internal controls are proteins that tell the cell when to enter each phase of the cell cycle. And a lot of these have stops or checks, like there's a stop from entering mitosis if the chromosomes aren't replicated. So the chromosomes replicate in interphase, specifically S phase, and if they're not fully replicated, or there's a mistake, if the cell can't enter mitosis. Otherwise, you get two mutated cells. There's another stop that stops the cell from entering anaphase until the chromosomes are attached to the spindle fibers. Because if not every chromosome is attached, then one of them is not going to be pulled apart, and you're going to have two cells with different number of chromosomes, which will be an issue. Okay? So a lot of the internal regulators stop the cell cycle until certain events have occurred. The external regulators are proteins that come from the outside, come from the rest of the body, and tell the cell when to divide, such as growth factors like HGH or human growth hormone. This stimulates cell growth and division, tells individual cells when to grow and divide. It's very important during embryonic development, and when you get a wound, it tells those skin cells, along with contact inhibition, that there's a wound and they need to start healing. Coming in contact with other cells again stops the cell division, so once the wound is healed, the cells stop moving. Okay. Very important external regulators to keep our body working functionally. Okay. If we lose control of cell growth, that's the basic definition of cancer, uncontrolled cell growth. So cancerous cells have stopped responding to the regulatory ability of our body, and they start forming tumors. They start growing more and more cells. They lack contact inhibition. They're growing without the external stimuli needed, and they just continue to grow. And if you just form a tumor in one area of your body that's not going anywhere, it's called a benign tumor, and it's not incredibly dangerous until the tumor breaks off and travels to other parts of the body. Okay? Things like skin cancer, the way they get dangerous is when it starts to metastasize and get to other parts of your body. A lot of them have a defective p53 gene. That's an internal regulator that controls a lot of the cell cycle. A big specific one is it stops metaphase until the chromosomes are perfectly duplicated. One of the many things it does. And so if you have that gene broken, then the cells can't control their cell cycle and they continue to proliferate, because that results in chromosome damage. More damage you have, the more mutations you have, the better the ability of the cancer to spread. Okay. The next set slides we'll talk about meiosis. Thank you very much.